Magnetic resonance imaging, or MRI, has emerged as a predominant imaging modality to localize transplanted stem cells and to better understand the mechanism of regeneration. These MRI studies typically use iron oxide nanoparticles as the imaging contrast agent. However, iron particles can remain in the tissue after stem cell death and can be taken up by macrophages, resulting in suboptimal conditions for tracking the viable transplanted cells. This video will demonstrate that manganese chloride is an effective alternative contrast agent. Labeling with manganese chloride offers several advantages over iron oxide labeling. For example, manganese gives a positive signal, whereas a negative signal is produced by iron particles. In addition, manganese ion is known to enter cells via voltage-gated calcium channels, so its appearance in MRI indicates viable, biologically active cells. Hi, I'm Mayumi Yamada from the laboratory of Phillip in the Division of Cardiovascular Medicine at Stanford University. Today, I will show you how to label human embryonic stem cells with manganese for magnetic resonance imaging. This procedure includes the following steps: labeling human embryonic stem cells with contrast agents, performing in vitro cellular magnetic resonance imaging, and analyzing the MRI results. So let's get started.